Good morning, everyone. I'm Janet Woodcock, and I'm delighted to be here, and I'm really excited to hear how far along you all have come. So let me step back a little bit. I was asked to talk about, what is the FDA? Why are we involved in this? What do we think about this? Well, when you think about rare diseases, and actually all diseases, but we'll talk about rare diseases this morning, and some of the challenges in getting products, getting innovations, new interventions to people with rare diseases. Of course, the first thing that comes to mind is there's a limited number of patients. And that's okay if maybe you're studying one thing, that's the only thing that's ever come forward. But when you have a lot of interventions you want to study, or you want to look at even what the standard of care is, often in rare diseases, the standard of care has just grown up. It's never really been tested. It's very empirical. You don't have a lot of data on it. And so there are many questions often you want to ask. And but with a limited number of patients, that becomes hard. And if you have more than one innovation you want to try out, then there may actually be competition for those folk. There are often few outcome measures, and they haven't been studied so well longitudinally, or there are a whole lot of outcome measures that each individual investigator, famous investigator, has used, and they don't fit together. And so then you're comparing apples and oranges and pears and so forth. And what has recently become really a problem is extrapolation. If you do a small trial with a small number of patients and you try to limit the characteristics of those patients so that you decrease variability and you improve your chances of finding a signal, well, then you may end up having only studied a certain people in that rare disease with certain characteristics. And then if the product is approved and on the market, then reimbursement may be limited to people with those characteristics. And I've been hearing more and more and more about that. Even if FDA gives a broader indication, the insurance companies may say, well, that type of person wasn't studied in the trial, and therefore. So that's a big problem. And also, most rare diseases, we have limited what we call mechanism and genetic understanding. It turns out if rare diseases have some genetic underpinnings, often they're highly variable, and that leads to a very heterogeneous disease course. And so we really need to understand the genotypes, the phenotypes, and then how that plays out over time to follow those people. And we often don't have that kind of often FDA will rely on that type of information to approve a new intervention if its disease is very small, compare it to external control group. But if that external control group, again, is apples and oranges, we don't understand the sources of variability, we don't understand why some people have a slower progression rate and others faster, and everything, then we're in trouble when there's controversies and so forth. So one of the things, yes, I freely admit, and still a risk here, but I've been one of the people really pushing platform trials and under that master protocols for quite a long time. And why is this? The biggest advantage to me, and this is why I think this community should really be interested, and others, is that these protocols and platforms their goal is to improve outcome of disease. Their goal isn't just to test one intervention or one kind of dose or this or that. It's really to progressively improve disease outcomes over time. And that is very powerful. That is patient-centered development of interventions and knowledge, right? So we will know what to do, what gives the best outcomes, and multiple things can be tested. So the other benefits, other than the fact that platform trials and master protocols are really focused around the disease rather than some intervention that you want to test, right? The other advantages are you can study multiple interventions at once. So you can have multiple arms. As was already said, you don't have just so many people on control groups because you can often use a single control group, and you can even use external controls. If they've been in the trial, for example, that makes it a lot easier. You understand a lot more. You're using the same outcome measures and so forth. But for example, the recent Ebola trial that was just completed, that was and declared victory and found 2 superior interventions to improve the outcomes of Ebola, had 4 different arms with different experimental treatments in it, okay? I mean, they were all run concurrently. They had the benefit, then they sort of, it was a bake-off, basically, and you see which ones resulted in better survival, right? And there were 2 winners, right? And that's great. Okay, so we understand now that that what we should use, but then we can go further. Okay, so another iteration of that might use them as the control group, and then you're going to try to do better than that. Can you add something on to them? Could they be used together? Would that help? And those questions then can be answered without waiting several years for those publications to be done and la-da-da. Now in Ebola, you have to the epidemic has to be ongoing, and if they manage to control it, they won't be able to do that for a while, but they can step in the next time an outbreak happens. So studying multiple interventions at once, you can see then, just from that small example, the power of that. You can generate natural history data as you go. And you can not only outcomes, but you can look at biomarker data. You can look at all sorts of information and learn instead of having that discarded, because then you can put that into your next iteration of the trial as you iterate the trial and build on the knowledge that you've gained. They can be adaptive, so you can seamlessly loop in those populations. Say you do a trial in a very limited, you do part of the master protocol in a very limited group of people because you want to decrease variability, because you want to see if there's any, hope there for this intervention. Okay, you find it. You find that it looks, it's better than whatever standard of care. Then you can broaden seamlessly into other populations to deal partly with this reimbursement, but the extrapolation problem, it is a real question. Okay, does it work in this additional population that is similar but slightly different? Does it work in this other population? Again, instead of waiting several years to mount another trial, maybe after the drug is approved in this small population, and then going through all sorts of struggles with insurance companies, to see if you can't get payment, then what you can do is have that data in hand. So that's really very helpful. And also there may be other questions. We never approve a drug where we know everything about it. All right, so there's always questions about dose, regimen, concomitant therapy. Those things can be seamlessly answered. Once you answer one question, you can simply roll into another question. And then common control is really important. I know for the many rare disease communities, I've heard from thousands of people with rare diseases, they don't want to be on the standard of care arm. They want the new arm because they aren't doing very well. We can minimize that by using the control for several other interventions. Once we get a successful intervention, we can use that as the control. So these things are very helpful, and you can do that much faster than you would do if you were developing, just doing a trial of a single agent that was centered around finding answers about that agent, not about, improving the disease. The lung map trial has had to get itself together, reconfigure itself several times because we approved drugs for non-small cell lung cancer, right? And all of a sudden those drugs had to be the control arm for a lot of the people because they work broadly. Or we approved drugs for a targeted subset, and all of a sudden that had to be the control arm for those subsets, just like you would see with Ebola, where you've seen a successful treatment. So the common control and the ability to roll into that is another thing. Now, these benefits, though, are are matched with challenges, and that's what you're here today, I think, partly to talk about. You know, in my long work in this world of government and everything, what amazes me is how difficult it is for people to envision something different than what they're doing now. It's just really hard, apparently. And what we're trying to do here today and talk about is not the traditional way of doing things. So every single player in the game is going to have to get their mind around doing something different. What I've learned, because I've done a huge amount of change efforts over my career, is people say it's resistance to change, and that really isn't what it is. It's like people can't imagine the world the way you're seeing it. We're seeing this platform trial, and we're seeing it operating, and we're seeing all these good results and everything, and people just see what's going to be a problem for them if they don't do things the way they've been doing, which have been, albeit, you know, taking a long time and everything, they have been successful and they're traditional. So bringing about change, and you have a large number of sectors you have to bring about change in, is very difficult. Other problems are there's a lot of upfront work. And we mentioned that, Alyssa Levange and I mentioned that in our paper. You have to do a lot more work to set something like this up and have it operate properly than you have to do a single trial, where people know how to do a single trial. Companies who are drug developers or device developers, whatever, they have this asset. They're used to controlling the asset completely. They like that control. It provides comfort for them. And so changing that, and they're losing some control going into a master protocol or platform, is very difficult. We heard from PIs here, so that's good. You know, the more people learn about this and the more it becomes more common, then the less the sort of barrier that people will have. But investigators often worry they'll lose control, okay? And there's this consortium, and and we have struggled with this in some of the trials we're doing, some of the platform trials, is people are used to, they want to study their own thing, they want to get in their paper in the New England Journal, right, and they want to la-da-da. And so that's a barrier. And then in Duchenne, actually, I've talked to some parents and so forth, and they're worried they will lose control because their belief has been, well, I can pick the best experimental trial for my child, and I can get my child into that trial. And we know, unfortunately, Billy and I probably know that, you know, the best people on Wall Street who like have millions of dollars riding on this, and they've been doing it for 30 years, they can't pick the winners. And the pharmaceutical industry still can't pick the winners. Otherwise, they'd have a higher success rate than 1 in 10, right, getting into the clinic. So, but if people really feel that they know, they have some inner conviction that this is the experimental product that their child should be on, as was said, this is going to be voluntary. It's not going to be, nobody will force people to get into a trial where they may be randomized to a variety of different arms of the trial, because none of us really have any idea what'll be the best for people. So the good news and the bad news is you're on the cutting edge, the forefront of really changing how products are developed. And I truly believe this will be ultimately the future, that we need to have a very strong infrastructure. We need to keep learning. The concept of continuous improvement needs to go into medical care. We need to continuously improve patients' outcomes, whether they have hypertension or whether they have muscular dystrophy, right? And that ought to be our focus, not on single entities like we do now. But, you know, there's this new entity and we're going to study that. We ought to look at all the disease outcomes, and are people doing better and better every year until we get to where they're doing just as well as if they were healthy. So that's the goal. And I wish you luck today. I think a committed community can make this happen. I truly believe it's the best thing for improving the outcomes more quickly and more effectively, and having more knowledge as you do it, but I don't underestimate the barriers. I will say, though, I think you have a good team here, and that, that they'll get it done. So thank you very much.
So good morning. I'm Billy Dunn. I know a lot of you. One of the great things about being engaged with the Duchenne community is I have a tangible reminder of that as I arrive today, and I'm not in the room for 30 seconds before I've shaken more hands than I can count, and I wasn't able to shake the hands of many more who I now know as colleagues and valuable colleagues, both in the scientific arena and in the general Duchenne community. We've had a lot of interaction, and that's going to continue. I'm really pleased to be here. I think you all know that I direct the neurology division at FDA. We're responsible for Duchenne. I have a couple other hats I wear at the FDA as well, but certainly here today we're focused very heavily on Duchenne. I really appreciate y'all inviting me to share a few thoughts. I echo already a lot of what Janet said, and part of the problem of having two opening comments is there will be some themes that are repeated, but I think you've heard themes both from the opening speakers, Dr. Woodcock and myself, that are important because we are in novel territory here. We really need to be careful about how we proceed because we need to do a good job. This is a tremendous opportunity. We're very invested in the success of these efforts. I have a number of my esteemed staff members here with me. They're scattered throughout, and they're going to be paying very close attention today to this discussion because I think one of the reasons I was talking to Dr. Larkindale about this, that it's important for us to be here, and one of the things you wanted my comments to focus on is kind of a little bit of that rubber meeting the road. We're going to actually do the work with our sponsors, with our drug developers, to get an effort like this up and operational. It's very important to the scientific community that succeed, and so we're here with our staff to make sure that we're setting off on the right foot. One of the first things I want to do is really congratulate everyone involved in this on the thoughtful, careful approach that has been brought to arriving at this meeting today. This is not the kickoff meeting. It's very apparent to me from my discussions with folks and from actual interactions I've had over the past couple of years. This has really been a long time coming just to get to this point. So there's been a tremendous amount of work already done. I know well how much work that represents, and I'm really pleased to say that. I think, Abby, you summarized a lot of what's gone into that very nicely. You also mentioned something, Abby, about the gold standard being a placebo trial. And I think as we're here talking about innovative approaches, I think we really need to think about that phrase gold standard. It gets thrown around an awful lot. I'm not sure, and this is probably going to get quoted and misquoted and misunderstood. I'm not sure that a placebo-controlled trial really should be called a gold standard. The gold standard, if there is one, is that we have interpretable, scientifically rigorous evidence that meets our scientific regulatory, which is really just good science, our threshold to legally be able to approve a drug. That's if there is a gold standard, that's what it is. There's many ways to get there, and there always have been. What the fact of the matter is, and I'm going to talk about this, is that a placebo-controlled trial in typical fashion is often the most efficient way to go about getting that. So it gets used. It's very interpretable. It has a lot of strengths. But I don't think we would worry so much about what that is as if we have to equal that. There's many paths to getting to what we call substantial evidence of effectiveness with a sufficient understanding of safety. That's what we need. And so this is a possible approach to do that. So I'm going to open up with, I don't know if I can have the next slide, or do I have control? How does this work? Somebody doing this for me? This? I went too far. There. So I'm going to talk for a little bit about control groups. I think when you're talking about a master protocol, platform trial, call it what you will today, one of the very important issues, and there are very many important issues when thinking about this, is what to do about the control group. So I'm going to spend a little time on that because there are so many complexities to setting up one of these, but this comes up frequently, often. It's a source of great desire for our developers and for our patients to say, let's minimize that in a variety of ways. Let's help our interpretability there. So I want to talk a little bit about that. One of the things that gets talked about a lot is the use of a historical control. This is a very challenging situation. Studies based entirely on a historical control can be challenging because the course of progression can be highly variable among patients, among individual patients. And there can be known and unknown differences among placebo cohorts, depending on which one you might choose. You can have changes in standard of care, as Abby mentioned. You had a slide about the evolution of this. You can have regional differences, population and genetic factors. All of these issues may contribute to this issue, along with unrecognized issues that you can't even identify. So when you're thinking about what to do with a historical control, which population you choose, is there more than one? Is there one gold standard historical control? What do you do with this? And how far removed temporally is it from the trial that you're doing? These things, that addresses the standard of care issue, and these issues can be very problematic. So as a result of that, results from historically controlled trials, purely historically controlled trials, are often difficult to interpret unless the effect size on a highly objective endpoint is relatively large and gives us great faith that it's differing from a reliable interpretation of what could possibly happen, historically. Something else to think about is this issue of placebo. Placebo is important in a lot of situations. It helps us identify, small but important effects. Small effect sizes, I'm not happy to say this, but it's what we deal with in neuroscience all the time. Small effect sizes is what we typically encounter. And though they're small, these are critical to identifying progressive neurological conditions. They're generally only detectable with some degree of placebo control. That's what gives us the sensitivity to observe these, and we don't want to fail to be able to identify these important small differences, especially when our drugs are proceeding technologically at such a rapid pace and allowing us to target fundamental mechanisms of disease such that small effects that we observe on the time of a trial, we have great hope that those may accumulate over time and result in substantial benefits for patients. So use of a placebo with equal randomization is generally the most efficient study design to rapidly arrive at an answer about a given drug's effect. That's just kind of a paradigm that's not unreasonable to say. But we also know that minimizing exposure to placebo and to control groups is very important. As we heard, that's a survey I think you administered, and that I think it was the number one cited reason, I think. Over many years, about reluctance to participate in the trial was, might I get a placebo? That's something we have to be aware of. If we can't conduct trials, then we can't get our answers. We can sit up there and kind of talk in some ivory tower about, we got to do the perfect trial. What we need to do is we need to get work done, and we need to do it efficiently, and I think that's what this effort's all about. So we have definitely heard about the desire to minimize exposure to placebo. That's to the degree that we can do that, we're very supportive of that. That's not a problem. Trial designs include features to identify a beneficial effect of the drug. We work on this very carefully, as early as possible in the planned course of any given study, whether it's a platform trial or not, to minimize the duration of placebo exposure. That's something that we really want to be on top of. We would hate to miss a benefit that becomes apparent earlier than a drug developer anticipated. Say they planned a 2-year trial. There's no way they'll see that benefit in 2 years. But what if we have people hopping up and doing well in 6 months? We want to be able to react to that. We don't want to just sit there and kind of wait for the trial to end. Another thing that we can do is we can use borrowing approaches to leverage historical control data in combination with concurrent control groups in order to minimize the exposure to concurrent controls. We can try to reduce the need for that by leveraging historical information, even in the setting of some of the complications that I described earlier. So while randomized, placebo-controlled, double-blind, gold standard studies, while these are typically the most efficient way to demonstrate definitively the efficacy of drugs, FDA unquestionably supports innovative strategies to expedite studies and minimize exposure to placebo. Master protocols, adaptive designs, enrichment strategies, these are all examples of things that we are supportive of and we want to work hard on accomplishing. Many of these things, in a subtle fashion, adaptive design is used constantly in almost all trials. Enrichment strategies are used subtly in almost all trials. Platform trials, master protocols, umbrellas, baskets, all the things in Dr. Woodcock's article. They're all kind of birds of a feather, and they're complicated. These are not things that just kind of get done subtly and, suddenly you find, I've got a platform trial, I didn't even know it. That's not how those work. These are tricky animals, and we really do need to work hard on them, and that's why we're having meetings like this. We're going to continue to do that. So how can a platform trial help? Well, from what I've said, the promise of a master protocol, which here is the intended result of the ongoing innovative work of this Duchenne Platform Trial Consortium, which, by the way, on the side, this Duchenne Platform Trial Consortium is wonderful because as you heard with our almost opening three executive member comments, it's a consortium of consortiums. You're able to leverage so much information from the accumulated experience of PPMD, IACT, and CPATH. It's impossible to overstate how important that is. There's a wealth of scientific and community knowledge here that can be leveraged. But the intended result of this ongoing innovative work of this consortium is to introduce efficiencies into the development of drugs for DMD while not losing the benefits of well-defined control groups and other aspects of clinical trial design that give us confidence in the results of any given trial. That's what we need to keep in mind. That's possible with this, and we have to get there. So one of the interesting things about being the FDA, and I say this a lot, we see everything, right? We can't talk about everything. We can talk about almost nothing, but we see everything. It's a very interesting place to be. And what that does is it gives us a lot of insight into what the real state is and what we can bring to the table. And I think that's one of the things, I was talking with Dr. Larkindale a little bit about this in advance of the meeting, and I think she emphasized to me that a reflection in some of my comments about what we're actually experiencing in the drug development space would be very useful here. FDA's experience with Duchenne trials, the numbers of interventions coming in, common areas of discussion, this is really important because we can reflect some of that experience as we contemplate how to continue proceeding with the development of this Duchenne-specific platform trial. Duchenne is a very exciting space because it's a real poster child for what I hope continues to be true in neuroscience. Dr. Woodcock and I have spoken often about how neuroscience is in just such a tremendously different place than it was years ago. Oncology had an explosion of development that was the result of increased scientific understanding. It used to be I'd get up here and tell any given community, well, what we need is more scientific understanding. We just don't know anything about our diseases. It's just a big, it's the depths of the ocean, it's outer space. We don't understand. The brain is complicated, the muscles are complicated, the nerves are complicated. What do we do? Woe is us. So what you did is you did your best and you did a lot of trials and you saw what stuck and you got lucky 1 in 10 times. But we have a lot of knowledge now, and Duchenne is just such a shining example of that. We have a tremendous understanding of the pathophysiology of this condition, of the mechanisms, and it's being reflected in our ability to target the disease in innovative and novel ways. That demands an ability to bring the efficiencies of innovative and novel study approaches. So what are the things that we talk about a lot with sponsors when they come in? Well, some of these things, and there is tremendous development in this space, as you know. One of the slides showed how many drugs are on that slide. It's not my slide, but there were a lot of drugs on that slide there, and there's even more than that. Again, we see everything. How do you deal with limited number of patients, both overall and especially with targeted subgroups? What do you do about that? That's a common theme for us. What is the choice of an appropriate control group, even with an individual trial? Endpoint selection and implementation, these are very important issues. How long should a trial be? What should its duration be? What is the adequacy of the disease characterization for any given sponsor's desired approach to studying Duchenne? How well do we understand the disease in a way that informs that trial's application? What should the size of the trial be, its powering? How are we incorporating, this is a critical one, it's so easy to say this quickly and let it slide by. How are we incorporating our awareness of evolving and dynamic standards of care into understanding what we want to do with a given trial? This is a source of great progress, and I'm happy about that. We're doing better for our patients, independent of drug approvals. That's great, but we have to factor that in. These things all tie together. I was talking about historical controls earlier, borrowing some of that information. We have to know what time period we're borrowing it from so we can understand that. As I said, we've listened to the patient voice on desire for fewer patients on placebo and inclusion of broader patient groups and trials. This is very important. And we know that we need an ever better understanding of disease progression, natural history, better comparator arms and endpoints. These are all issues that we talk about regularly with sponsors and that we're very aware of. So a platform trial has great promise in addressing these issues. It can address very many of them. So, kind of coming to the punchline, although I have some more comments, Jane, we talked, and one of the things that I think there was some fear of is, well, there's an article out there, and we have some lip service maybe to platform trials, but what's really going to happen when people come talk to us? And I mean, I'm happy to spill those beans. We are supportive, but we're not going to shy away from the complexities. These are very, very challenging. The upfront work, the ongoing work, the maintenance, but the allure of doing it is extremely high. The ability to create a dynamic, living trial that moves along with the clinical care and the clinical research is a tremendous opportunity. So the punchline is, yes, we are ready to work on these. We are working on these. You already heard some mention of some other areas in the neuroscience space where we're working with other developers and third-party agencies to get platform trials off the ground. They're coming. They're coming. We're on the cutting edge, and we're very happy that you are too. We're all here together. So platform trials can address some of these things, and this is an opportunity to talk a little bit about data standardization and sharing, something that's critical in a platform trial scenario. Platform trials need data standardization so that the various moving parts of the platform trial can speak to each other. And these platform trials lead to enhanced suitability for data aggregation and contribution to data sharing efforts, something else that I'm absolutely passionate about, that I work very hard on. Many of you in the audience know this is a project that I am deeply involved in, data sharing. Jane knows this very, very well. I have the great privilege to work hard on co-leading an effort with the Critical Path Institute focused on drug development, specific focused neuroscience data sharing. It's very critical to do this, to inform and advance drug development for all diseases, but especially for rare diseases. We have an upcoming effort that's not the focus of this meeting, but many of you are aware that we have an upcoming effort that FDA is directing to specifically accelerate drug development for rare diseases. This is going to kick off next week. Platform trials will complement those efforts. Data sharing is not only relevant for the platform trial in relation to the larger scientific community, but it's a critical concept within the platform trial itself. One of the great advantages you have in your platform trial consortium is the presence, as I said, of this aggregated expertise of it's a consortium of consortiums. And the Critical Path Institute, which Dr. Arkendale comes from, I just can't tell you how much experience they bring to the table in terms of data aggregation, data quality, standardization. You're already way down the track in terms of getting this platform trial where it needs to be by having brought people with such expertise to the table. Something else to think about in platform trials is precision targeting. Platform trials become particularly attractive as the targets for new drugs become more precise. DMD, as I said, is an excellent example of a disease where our knowledge allows precision targeting and supports the pursuit of a master protocol approach. Our scientific knowledge of the disease gives us that ability. So is Duchenne suited for the application of a master protocol? Clearly, my comments suggest that it is. Platform trials are particularly well suited for graduation approaches, one might call them, which rely heavily on the knowledge from well-characterized diseases, allowing the use of biomarker-informed assessments for mid-phase go, no-go decisions, with the potential for individual drug-specific trials post-graduation. There are additional complexities when contemplating a platform trial intended to provide definitive evidence of effectiveness, but Duchenne is an excellent example in the neuroscience space of a disease that may be able to benefit from both early and later phase master protocol approaches. I would encourage the community and the committee, as we're thinking about how to get this off the ground, this has a real opportunity to be a living protocol where, given our ability in Duchenne to understand the disease and to recognize the role that dystrophin plays in it, our openness to the use of dystrophin as an endpoint to potentially support what we call accelerated approval, with the need nonetheless for subsequent clinical investigation to confirm what we hope is the benefit of the drug, you might have an opportunity here for a real novel approach where a master protocol could actually evaluate some of those early phase biomarker-based assessments and allow for rolling into confirmatory trials. You could have not only these early phase, but late phase issues all wrapped up into one thing. That requires a tremendous commitment from the patient community, from the scientific community. Egos have to be put aside. Equipoise has to be maintained. Organization has to be paramount. But you can do this, and if we can do that, we will be able to churn through the evaluation of promising therapy after promising therapy and identify quickly those that are best for our patients with Duchenne, which is what we all want. So I think Duchenne is particularly suitable for this. There are many, many logistical issues associated with the master protocol, many unique aspects. They require thoughtful and detailed advanced planning and discussion. But successfully addressing these issues is at the heart of successfully achieving the efficiency that master protocols offer. Infrastructural issues are crucial. Common screening procedures, common governance procedures with a centralized IRB and centralized steering, adjudication, and data monitoring committees, single entities, not multiple dispersed entities. Fewer resources than therefore are needed. Uniform and consistent approaches and decisions are applied to the master protocol, and you have increased efficiency, which is really what this is all about. Use of a clinical trial network. I've been saying for many years, and Janet, I know we've talked about this a lot. As we move towards what we're working on in the neuroscience space at FDA, I really do believe, both in the rare disease neuroscience space and in the larger neuroscience space, that moving our neurology field into a recognition that research participation is part of clinical care and having broad networks set up is going to be key to this. The Duchenne community is already well poised to accomplish this, and it needs to be a defractured networked approach. Central randomization, uniform quality control efforts are advantages of the platform trial. Logistics have to be applied to that. The logistical considerations have to be taken into account. But you have the opportunity to feed back in those QC measures and affect all the different aspects of the trial all at one time in kind of a net or a web, much more efficient. There's also trial design issues to consider. We have already discussed many of these, but there's various adaptive design features that can be used, common controls, as I mentioned, including issues related to the borrowing and incorporation of natural history data. Unique or less common statistical approaches require careful consideration, but that doesn't mean they might not be acceptable. It just means they have to have careful discussion and a lot of front-loaded thought. So there are many advantages that make this large upfront effort worthwhile. More inclusiveness in enrollment, reduced startup time to investigate a new agent, more patients on the active drug without diminishing the rigor of the study, a greater opportunity for continued participation in various trials, not just one. How frustrating it is to have that exciting moment to participate in your trial, have it end, and what else can a patient do? Across the span of a progressive disease where patients worsen until we find the cure, where patients worsen, we want to have an opportunity for research across that span of progression. So there's great opportunity there. Ideally, the heavier lift at the outset of starting one of these platform trials provides a net increase in efficiency over the lifespan of the platform trial. And I'm particularly excited about the fact that I think of a platform trial as a living entity that moves along in contemporary fashion with evolving care and with evolving therapies that are being developed. That has tremendous implications that we don't have time to go into here, but one of the things that I spend a lot of time thinking about is once we get over the hurdle of how to deal with external controls, what do we do as they become more temporally remote? Having an ongoing living trial allows us to maintain some contemporaneous aspect to our control group, even in a platform situation, which makes it much, much more interpretable and helps us wrestle with that evolving standard of care issue. Finally, what does one of these need? It needs a champion, and that's really what y'all here are. All right. A successful effort to develop a platform trial requires an organization to lead the pre-competitive effort and make it acceptable for the various development programs that all want to benefit from what it has to offer. There's a lot of issues at play there. You know, a lot of thoughtfulness about control and who's willing to give what, but those can be overcome. This consortium is clearly putting in the necessary work to achieve this goal. I'm absolutely delighted that we're able to be a part of it. And I really appreciate the opportunity to have shared my thoughts on this. It makes me even more excited saying them out loud. So thank you very much. Again, this meeting is going to be a great one, and I appreciate the opportunity to be here. Y'all take care.